Welcome to this live from the field event on the topic of microbes, both those that cause sickness and those involved in symbiosis, where we will learn about cutting edge research and teaching strategies taking place in tropical forests spanning 3 continents. My name is Carrie Winninger, and I'm the director of Sonoma State University's Center for Environmental Inquiry. You can learn more about us at cei.sonoma.edu. This Live from the Field event is a collaboration between the Organization of Biological Field Stations, OBFS, and the Ocelots Network, online content for experiential learning of tropical systems, with support from the National Science Foundation. For instructors in the audience, be sure to take a look at the document that I'll be pasting in the chat called Ocelots Modules and Implementation Plan Links and pre-event reading. And please also visit thevirtualfield.org to view past events and access other virtual learning experiences for college students created at field stations and marine laboratories. Here are some guidelines for the webinar. All participants are muted and your video cameras are off. I do see the chat is disabled and I'll be trying to fix that soon. Please use the chat button when available to communicate with us during the event. And remember to choose all panelists and attendees so we'll all see your comments. Please submit questions that you have for the presenters using the Q&A button, not the chat. If you're here as part of a college class, it's really important to make sure your full name is visible so you receive credit for attending. So make sure either your name is in your username or you sign it at the bottom of a question that you type into the Q&A. Lastly, we will be providing information about 2 surveys, 1 survey for instructors, 1 for students, and we really hope you'll be kind enough to complete those for us. All right. Today, we are fortunate enough to have as our moderator someone with a foot in both the field worlds of field science and education. Dr. Jeff Clemens is an associate professor of biology at Thomas Jefferson University in Philadelphia. Jeff did his graduate and postdoctoral research working on aspects of forest regeneration in the Area de Conservación Guanacaste in northwest Costa Rica. He continues to conduct fieldwork in Guanacaste and the temperate forests of Pennsylvania, and is the co-author of the Ocelots module, Snapshot Serengeti. So without further ado, I will turn this over to Jeff.
Hi, everyone. Thank you all for joining us. So we welcome all of these classes, faculty, other participants to this Live from the Field event. We're going to take you on a virtual field trip today to 3 research sites around the world. And as you'll see, we're going to have a chance to visit virtually with professionals at those 3 different locations, all of whom have joined us here today as panelists. So Live from the Field events are a project of the Virtual Field, which is an international coalition of 50 field stations and marine laboratories. The virtual learning experiences help college students build observation, communication, questioning, and data analysis skills through the use of 360-degree videos, underwater films, and many more exciting things. So if you want to learn more about any of that, check out thevirtualfield.org. As Carrie mentioned, this particular Live from the Field has been developed in close collaboration with the Ocelots Network, which she also mentioned that I'm a part of. And that's a group that brings together researchers, active learning pedagogy specialists, software developers, and media specialists to create an open access online resource library of learning and teaching modules in tropical biology. For those of you joining us from Jefferson, you might have experience with one of these if you've taken our ecology class. The goal of these modules is to get excitement built up in undergraduate courses around the idea of field biology, and to broaden your cultural and geographic perspectives, and enhance some of your reasoning and quantitative skills when it comes to working with data. So today we're going to take a look at projects that deal specifically with the world of microbes. And you might think about microbiology as something that only happens in the lab. You think about people in white coats interacting with creatures that are growing in little Petri dishes. And we hope that at the end of this, you'll realize that you couldn't be more wrong, and that field-based investigations of microbes are just as important and dynamic as the field-based studies of plants and animals that you might be more familiar with if you think about field biology or just watch them in nature documentaries. So today we're going to be taking a look at the extreme ends of the spectrum when it comes to the interaction of microbes with the so-called higher organisms. At one end of that spectrum is symbiosis, the essential interactions between microbes and other organisms that allow groups of organisms as diverse as lichens, trees, and us, human beings, to efficiently obtain nutrients from our environments. At the other end of the spectrum is the role of microbes as pathogens, organisms that inhibit the growth and survival of species that they parasitize. I'm very excited to serve as the host for this event, as my postdoctoral research actually focused on the ecological importance of a particular group of microbes that I know Lindsay's going to talk about, the mycorrhizal fungi, in forest restoration in a kind of tropical forest called tropical dry forest. Um, so I got involved with all this with ocelots to share the excitement of fieldwork in the tropics with students who might not get a chance to visit the tropics themselves, or who might not have had a chance to visit them yet but plan to someday. So I'm excited to present these three teams of biologists to you. Two of them have created modules based off of their own field research, and the third has implemented one of these modules in their own classroom with their students, and they're going to talk a little bit about that experience. Each team has prepared an 8-minute video describing their work and experiences. And after the videos, they're going to be available to answer your questions live. So I encourage everyone to post your questions in the Q&A as we go along. Don't wait till the end to post your questions up, but post them up as soon as you think of them. And Carrie and I will gather them up and ask them out loud to the presenters during the panel discussion portion at the end. So if you're a student, also please let us know that in your question. Flag it and say, student question, and we'll prioritize those student questions first. Sorry, lost control of my mouse there for a second. Our journey is going to start. Our first video that we're going to see is in a place that's very near and dear to my heart, both conceptually and geographically. It's the La Selva Biological Station in Costa Rica. And La Selva is one of the most important field stations in the history of tropical biology. And it's been the venue for a lot of students' first introduction to the tropics, as it has hosted many courses that are run by a group called the Organization for Tropical Studies. It's also been the site of a lot of long-term research work in tropical ecology and forest dynamics. So Lindsay McCullough and Jorge Mora Cibaja they both work on the relationship of soil microbes to natural forest regeneration processes. Their ocelots module is called Plant Microbe Mutualisms in Canopy Gap Recovery, and focuses on how microbes contribute to forest regeneration following disturbances such as tree fall gaps in the forest canopy.
Sorry, folks, looking like I'm having trouble with this video. All right, well, let's try it a different way then. Always have to have trouble right when you are presenting, right? I promise it's a very cool video and worth waiting for. Let's try this one more time. Share my screen. All right, hopefully this one works. I see it. It is bound to work this time, everybody. Here we go.
Hello, my name is Lindsay McCullough, and I'm an assistant professor of teaching at the University of California, Riverside. Partners are all working together to affect ecosystem level processes, like how carbon moves through ecosystems, or nitrogen moves through ecosystems, or how they might influence what plants survive, when, and where.
Just starting over again because you couldn't hear it.
Microbial partners are all working at the Selva Biological Station in Symbiosis, Costa Rica. And this is where a lot of my research has taken place. I teach courses about intro to environmental sciences, plant microbial interactions, plant soil interactions. So I like to think a lot about how plants and soil and their microbial partners are all working together to affect ecosystem level processes, like how carbon moves through ecosystems or nitrogen moves through ecosystems, or how they might influence what plants survive, when, and where. My career as a researcher started when I was an undergraduate. Before then, I didn't have a lot of experience spending time outside or traveling a lot, so I got an opportunity to come here to Costa Rica with Dr. Catherine Cardelus and Dr. Carrie Woods to study the plants that actually live in the rainforest canopy. So these plants that live on top of other plants called epiphytes. You know, I didn't have a lot of experience traveling. I didn't have a lot of experience working in a place that looked like this, where it's hot and humid and there's insects that are huge. And so it was a little uncomfortable at first, but now it's a place that I've grown.
My name is Jorge Mora Cibaja. I'm from Costa Rica, and I am a co-author in this module. Currently, I am a PhD student at the University of California, Riverside, at Matt Frederick Lab, where I am trying to understand how environmental factors shape the microbial community composition between both plants and bees. My journey in science started in 2018 when I started to work monitoring biological corridors to better understand their functional connectivity around Costa Rica. Few years later, I get involved in this project where I start to understand more about how disturbance influence the dynamics in the rainforest. And then I start to get so excited about the idea of create this module because one of my aims as a scientist is make science more accessible for people from Costa Rica and other countries.
One of the reasons why I wanted to develop this module in particular, thinking about plant and microbial mutualisms, is because plant microbial mutualisms are actually globally distributed. So it's become very popular in popular science to talk about mycorrhizal fungi. So mycorrhizal fungi form relationships with most vascular plants found around the world, which means that these fungi that grow into the root systems of plants and into the soil are really essential for how plants globally get the essential nutrients that they need to grow. So it doesn't really matter if you're in a tropical forest like this or in a more temperate zone. These interactions are really important for our students to understand. And additionally, there's another plant microbial mutualism, nitrogen fixation, which occurs between plants in the Fabaceae family or legumes that form these nodule structures on their roots. And they have these bacteria that grow in there called nitrogen fixing bacteria. And those bacteria take nitrogen from the atmosphere and turn it into a form that plants can use. And so really important agricultural plants like soybeans do this, but there are also really large tropical forest trees that do that. And so understanding the dynamics of how plants rely on microbes in the soil and in their roots to acquire the essential nutrients they need to grow and survive is something that is a really important topic across ecosystems. So when I was developing this module, I was based in the northeast of the United States in temperate deciduous forests. And there we also find those interactions, but happening at a lower diversity of plant species for nitrogen fixing bacteria. So there is a high diversity of plants in the tropics that form this relationship with nitrogen fixing bacteria. And that is something I really wanted to highlight, even though this relationship is important throughout the globe. So that's one of the major differences when we think about some of the ecosystems in the northeast of the United States, where I was based at the time, compared to the tropics, is the diversity of plants and animals and all sorts of organisms that we can find here in the tropics compared to in some of our temperate ecosystems. But another really important part of the tropics, something that I always find really fascinating to think about, is often when we think about a tropical rainforest, we think about this pristine forest with these big, beautiful trees. And that is what exists here, but also another really common and important part of the ecological dynamics of this ecosystem is disturbance. So here you can see a really large tree fall that happened about a decade ago. And so these large trees, when they come to the end of their life, fall. And they create this really big opening in the canopy that allows light to reach the forest floor. So that's another big difference that we have between the tropics and temperate forests, is often light is a limiting resource here in the tropics when you're on the floor, because the canopy is so big here, very little light reaches the ground.
Through this model, we hope students will develop a better understanding of the role of disturbance in forest dynamics, build their own hypotheses, and learn how to interpret figures from primary scientific literature.
So I think some of the most surprising things about this module for students is that disturbance is really a key and important part of these ecosystems. A lot of times we think of a big tree fall or a disturbance as something negative that has happened, but it's actually a really important part for how these forests regenerate. And also the concept of how plants are really not these solitary organisms out here in the forest working alone. They have these really important close mutualistic relationships with microbes that are essential for how they function, grow, and develop into these ecosystems. And that often these plants are partnering with more than one microbe at the same time. So plants have these different demands on their valuable resource of carbon to gain different nutrients like nitrogen and phosphorus, which are essential for plant growth. So I think that these concepts are really key to tropical biology, to ecology, how dynamic ecosystems are, how there are many different organisms interacting, and the context dependence of that. I hope that students take from this module that there's a lot of creative ways that we have to use to ask these kinds of questions. So building mini shade houses and putting them out into the forest, using really great tools like isotope tracing to understand how nutrients are moving through our ecosystems and understand how plants are making a living with their mutualistic microbial partners to create these ecosystems that are incredibly dynamic and diverse. Thank you.
Being in the middle of the rainforest is an incredible experience. The abundance of life, the sounds of birds, and the towering trees intertwined with lianas are amazing. Looking at the canopy is incredible to realize that all of this is possible because of the processes happening all underground. And actually, that's my favorite part of this model. By the end, students gain a deeper understanding of one of the main reasons behind the extraordinary biodiversity of this ecosystem.
Okay, now we're going to travel to somewhere that is probably one of the first places that comes to mind when you think about the tropics, which is the Amazon rainforest. And this module also starts with someone who you might not think of as the most likely field biologist, which is a medical doctor, which is probably something that many of you are studying to be someday. This work, exploring the role of microbes as pathogens, was conducted and turned into an Ocelots learning module called Oropuche Fever in the Americas, Discovering the Mystery of the Virus Behind an Emerging Zoonosis by Dr. Carlos Galler and Daniel Romero Alvarez, a medical doctor who also holds a PhD in ecology and evolutionary biology, so a very unique perspective. Their work focused on how ecological changes to landscapes contribute to the spread of this novel zoonotic virus. And zoonotic means a virus that spreads between animals and humans. And their module uses the techniques of ecological and epidemiological modeling to predict where Oropuche may be found in the future.
Hi, my name is Daniel Romero Alvarez. I work as an associate professor and principal investigator at Universidad Internacional SEC in Quito, Ecuador.
All right, apparently we cannot see the video, so I'm going to try this again. I'm going to put in the chat some links that you all can explore while I'm doing that. Better? Thumbs up, Carlos? Can we see that video now? All right, let's start this over.
Hi, my name is Daniel Romero Alvarez. I work as an associate professor and principal investigator at Universidad Internacional SEC in Quito, Ecuador. I'm a medical doctor turned disease ecologist. After obtaining my MD, I decided to pursue a degree in ecology and evolutionary biology to do research on the origin, distribution, and spread of infectious diseases. Ecology of infectious diseases is still a relatively emerging topic. It has become very popular after the events of the COVID-19 pandemic between 2020 and 2022, and the rising popularity of concepts like One Health or planetary health. This field of science puts effort on understanding the factors determining where, how, and why parasites such as bacteria, virus, helminths, fungi, prions, protozoans affect animal populations, wildlife, or humans. I deeply enjoy this discipline since it allows me to understand infectious diseases in a holistic way. It is never only about the human case. It is about deforestation driving changes in wildlife populations that increases human-wildlife interactions that determine larger opportunities for parasites to start epidemics. The best science is a result of a collaborative effort. Luis Escobar is one of my most important mentors in my scientific career. A long time ago, I saw his presentation on rabies in cattle in Chile, and I instantly fell in love with the topic, and I knew that I wanted to follow this path of knowledge. Luis always supported me and helped me to pursue my PhD. We started to work on Oropouche virus back in 2015.
I am an associate professor in disease ecology at Virginia Tech. I study diseases affecting fish and wildlife and how the landscape, the temperature, the disturbances can affect or influence the presence of these diseases in the wild. I can show you the office. This is where I work. I have a shrimp tank. Then we have the campus of the university. I'm going to take you really fast to see the lab. This is Professor Weisenberg working during his sabbatical. This is where the students work. This is the lab. We have them when they're not working, they're eating. So we have the rest of the lab here.
For this particular effort, we have joined forces with Carlos Goller.
Hello, my name is Dr. Carlos Christopher Goller, and I'm a teaching professor at North Carolina State University in the Department of Biological Sciences. My discipline is microbiology, and I have studied how microbes are around us and how we can use sequencing technology to learn more about the diversity of microbes in and around us.
Oropouche virus is a 3-segmented RNA virus. This means that the genome is divided into 3 sections. The small, medium, and large segments are crucial for virus development. In 2016, 61 cases of Oropouche fever were detected for the first time in the department of Cusco, Peru, South America, and thanks to the application of landscape analysis and free satellite imagery, we were able to suggest that vegetation loss might be one driver of the outbreak. For the Amazonian region, we are working at Limoncocha Research Station, which is a protected natural reserve in the province of Sucumbíos, Ecuador. Here, we are using systematic approaches to collect mosquitoes throughout Limoncocha town to characterize the species of vectors living in the area to later apply molecular techniques to identify the virus. It is amazing to see how even in the most unexpected collections of water, we are able to recover mosquito larvae. In the Andean region, we are still defining collection sites. It was unexpected that the most recent cases of Oropouche fever reported in Ecuador were detected at high elevations. Usually, you expect to find cases of arbovirus disease in the tropics. For Ecuador, this is either the coast at the west of the Andes mountain ridge or the Amazon at the east.
Recently, we just published a new article regarding Oropouche virus. We found a case in the province of Bolivar in the canton of Caluma, here in the Andean region of Ecuador. And we used published genomic data in order to answer one question. Was this strain similar to the one circulating in the epidemic of Oropouche virus in Brazil? And the answer was that the strain located here in Ecuador was endemic. It was not related with the epidemic. And that is super important because with this publication, we confirmed the circulation of Oropouche virus across the country. And let me share a very important piece of advice for this state of your career. Because we are surrounded with information, don't overestimate the power of secondary data. There are so many data sources that are open and freely available for you to put together an interesting research. My first publication on Oropouche virus as a first author was done entirely with data downloaded by the Ministry of Public Health of Peru because they decided to share it with everybody. We download that data, we download satellite information that is actually also free, and we characterize the landscape that allowed that particular outbreak. Don't forget to use secondary data. By that way, we will be able to answer interesting questions very quickly. Research is not at all a linear process. There are a lot of sources of information that will enter into your world of ideas, and you will have to take a lot of information from there to put together a strong hypothesis. Like when you are creating a song, you don't start from the first paragraph and then you go to the chorus and then you end the song. Usually melodic ideas come from different parts. And then it's the job of the researcher to put together those pieces to have an entire puzzle, which is your research question.
Vámonos al inframundo y quemarnos hasta despertar.
All right. Our last stop will be in Bengaluru, India, where Professor Arindam Mitra at RV University used Lindsay and Jorge's module in the classroom with his undergraduate microbiology students. He then authored an implementation plan for other instructors who would like to use this module in the future. In this video, Dr. Mitra discusses how he got interested in microbiology, how he uses the ocelots module in his classroom, and what he thinks students take away from using these kind of learning modules in terms of both the microbiology content that they learn and the development of their scientific reasoning skills.
My goodness, Zoom, you are not happy with me today. We're going to try this again.
Hello, everyone. I'm Arindam Mitra, currently a professor and dean in the School of Allied and Healthcare Professions at our university, situated in Bangalore, Karnataka. I've completed my undergraduate degree in pharmacy and master's in biotechnology from Jadavpur University, India. I also completed my doctoral studies from the University of Maryland at College Park, followed by a postdoctoral research at the Arizona State University in Temple, in the United States. I'm currently teaching microbiology to undergraduate students enrolled in various programs under the School of Allied and Healthcare Professions at our university. I have previously taught courses in microbiology, cell biology, recombinant unit technology at both undergraduate and postgraduate levels across the microbiology, biotechnology, and biochemistry programs. I enjoy teaching microbiology because microbes are ubiquitous and play a key role in our planet's ecosystem. While they often make headlines for their role in causing disease or promoting health, microbes also play a vital role in maintaining environmental balance by cleaning ecosystems and recycling nutrients through various biogeochemical cycles. My first exposure to microbiology came during my master's project, where I investigated outer membrane protein mutants to assess fast susceptibility in specific Escherichia coli strains. Routine troubleshooting was key to overcoming setbacks and ensuring steady progress. Keeping detailed, updated records enable us to track both successful and unsuccessful approaches, providing a clear roadmap of the project's evolution. Incorporating experimental strategies informed by literature and data-driven insights was crucial to advancing the research. My teaching philosophy is grounded in both my research background and formal training in microbiology. I actively engage in various professional development activities to stay current with best practices in pedagogy, which I regularly integrate into my teaching approach. I have been teaching introductory microbiology and biochemistry courses to undergraduate and postgraduate students in India. The ocelots, or the online content for experiential learning and tropical system modules, are particularly relevant in the Indian context, given the country's biologically rich tropical ecosystems ranging from the rainforest of the Western Ghats and Northeast India to the coastal tropical belts. The module, Plant Microbial Mutualisms in Canopy Gap Recovery, examines how soil microbial communities facilitate forest regeneration following disturbances. This is highly applicable to Indian ecosystems where canopy gaps frequently result from cyclones, tree fall, selective logging, and other anthropogenic pressures. Understanding the role of microbial mutualisms in forest recovery can directly inform reforestation and ecosystem restoration efforts in India. The module's emphasis on microbial responses to ecological disturbances is especially pertinent given India's ongoing environmental challenges such as deforestation, mining, shifting cultivation, and industrial pollution. Insights into microbe-plant interactions, soil microbial threats, and nutrient cycling provide critical understanding of soil resilience and vegetation recovery in tropical regions. Students' perception shifted from viewing forest recovery as a universal process to understanding how factors like monsoon seasonality, human disturbance, and local biodiversity shape plant microbe interactions in India. They moved from passively learning to critically evaluating the module's applicability, recognizing the need for region-specific ecological models. This comparison made the concepts more relevant and encouraged deep learning and engagement with local environmental issues. To make the module relevant for Indian students, the students were instructed about region-specific elements such as monsoon-driven seasonality, common local disturbances like shifting cultivation in fire, and native tree and microbial species. This adaptation helps students relate more deeply to the material and apply it in the local environmental context. Students' new perspectives foster systems thinking and context-aware reasoning, helping them understand how ecological interactions like plant microbe mutualisms vary across regions. This prepares them to apply scientific concepts more critically in fields such as agriculture, climate science, and restoration ecology. It also strengthens their skills in data analysis, experimental design, and interdisciplinary connections. The most surprising part for students was learning how crucial underground microbial networks are in helping tropical forests recover after disturbance, an often overlooked but vital part of ecosystem resilience. They were particularly intrigued by the idea that mutualistic microbes like mycorrhiza and nitrogen-fixing bacteria can significantly influence which plants survive and thrive in canopy gaps. This challenged their assumptions that forest regeneration is driven mainly by above ground factors like sunlight and seed dispersal. The module helped students see that ecological research involved forming hypotheses, adjusting methods, and interpreting complex, sometimes unexpected results. By following real-world studies of plant-microbe interactions in canopy gaps, students understand how field conditions and data variability shape scientific discovery. This gives them a more realistic view of research as an ongoing, iterative process rather than direct answers. Students may apply their new perceptions by approaching scientific research with considerations for complexity, context, and the role of hidden factors like microbial interactions. They will be more inclined to question assumptions, consider local variables, and embrace the trial and error nature of research. This mindset will improve their ability to design thoughtful, adaptable studies across diverse scientific fields.
Okay, just another reminder, everybody, keep pushing us some questions into the question and answer panel that we have. Just in case you notice, down at the bottom, there's a chat window on my Zoom, and then there's a separate Q&A window for posting questions into the chat. So make sure you're going over to that Q&A one so we can get some collected. Before we jump into that round of Q&A, we do have a favor to ask you all, and we're going to show you a slide here that's got some survey information on it. That information is important for us to gain insights about your experience in this webinar, both for our reporting back to our funders and to help us improve these events going forward. So if you're an instructor, please keep an eye out for an email about the consent form and the post-event survey. It's going to come to you from our external evaluator, and her name is Liesl Sariak, and that's the Measurement Resources Company. So that consent form needs to be completed before you can do the survey. Right now, we currently have a link for the student survey is sitting in the chat. And do we have the slide to come up here, Carrie, for the with the QR code on it?
Yeah, so I think I'm sharing my screen right now. Can anybody else see that? It says post-event surveys on it. Danielle, was that a yes or a no? Thumbs up or thumbs down?
That was a no, I cannot see your screen. It's just a black screen.
Interesting. All right, we'll stop sharing and start again. Zoom is having a fun day today.
So we hope you'll take this survey, not right now, not immediately, but right after the event. We hope you participate in the Q&A and kind of focus on that. And we really want your feedback after the event is over. But the URL you can see for the survey, if you want to grab it and save it, is down in the chat. And here we are. Here is our survey slide. And so our student survey, if you want to take a picture or a screenshot of that QR code as it's up right now, and then fill out the survey after we're done with the event.
All right, so take that photo now. There's also that link in the chat.
And it should just take you about 2 minutes. It's a very quick survey.
Yeah, but please wait until after the event. So let's get going with the panel discussion. If everybody wants to turn their video and audio on, go take it away, Jeff.
Okay, so now it's time for your questions for the panelists. As promised, please keep posting your questions for the presenters using that Q&A button. Some of the college classes here have a time constraint of 50 minutes, so we're going to prioritize student questions coming in. After that, we'll answer questions from other attendees and hang around for another 10 minutes or so. So the panelists are going to try and keep their answers to a minute or less, reminder, so that we can get to as many of these questions as possible. So Carrie, why don't you take it away with the first question?
All right. So to get us all started, I would like to hear from all of the presenters briefly on the same question. So let's just go in the order that we met you all. So we'll start with Lindsay, then Jorge, followed by Carlos, then Danielle, and finishing up with Arundam. Remember that order? Great. Okay. So that very first question is, please tell us or remind us what drew you to field research or microbiology in the first place?
Hello, I'm Lindsay. And so what first brought me to doing field biology was a professor in my class said there was an opportunity to go to Costa Rica. I'd never left the country before. It sounded amazing. And basically La Selva is like summer camp for scientists. So it was so much fun. And I met so many amazing people that when I learned I could do this for my whole life, I was sold.
I'm Jorge. Actually, I can activate my video because said that I don't know that I don't have permission from the host, but that's work like in the field actually, like kind of like the same experience like with Lindsay. I took this course like biogeography actually, and I was biking to the university and this professor asked me, "Hey, do you have like a good physical condition, right? Because you're always biking." And I said, I guess so. So this guy just offered me like a position like climbing trees to place cameras. Then like my journey started, like kind of like a random thing, but I start to love, and I'm so passionate about like ecology. But I start super random, and I'm here doing my PhD.
So I'm Carlos Scholar, and I love playing in the lab. But I've always, since I was in high school in Mexico, I really loved infectious diseases and learning about viruses. And while the lab work is awesome, I need opportunities to connect with others who do field work and can bring the field work into the classroom and connect it to disease surveillance and looking at the evolution of infectious diseases.
Hello, everybody. My name is Daniel Romero Alvarez. I'm working here in Ecuador, and I enter into the field going to the field and actually performing experiments there from the parasitology. So for me, because I am a medical doctor, clinical life was kind of incomplete in terms of infectious diseases because patients were coming with their particular illness from somewhere. And I was having the idea of going to the place where they were actually getting the disease to see if they were aware to stop the pathogen to actually starting the disease in the first place. So that was my origin. And I started doing research work with parasitology, looking for parasites, gastrointestinal parasites here in Ecuador.
Hello, everyone. My name is Arundam Mitra, and I'm currently in RV University as a Dean of School of Allied and Healthcare Professions. It's a new school. We are starting with programs like all our undergraduate programs focusing on healthcare aspects like cardiac care, anesthesia, and operation theater technology, as well as medical laboratory technology. So my initial, attraction to the field of microbiology came during the master's program when I worked on a fast susceptibility to certain E. coli strains. So we are working on outer membrane proteins. And since then, I got involved in various projects focusing on two-component regulatory system, looking at virulence, working with avian pathogenic E. coli, then with uropathogenic E. coli, and also the laboratory strain of E. coli. Later, I moved on to developing vaccines using Salmonella gallinarum, particularly, which is a disease of the poultry.
affecting significant loss of poultry by this disease. So we developed vaccines to curb this disease or reduce the burden of this disease. And later I moved on to much more much more on applied part, mainly focusing on the biofilms, wastewater surveillance, and evolution of infectious disease. So a lot of broad area that I'm currently working on. Thank you.
So for those students who have to leave now, I just wanted to say thanks for joining us, and that we'll be emailing out follow-up information, including questions we didn't get to. But for everybody, if you can stay, we're still going to go through and answer some more of these questions, but just stay tuned for more info for those who are not. All right, I have a question for Lindsay, who is masquerading as Daniel Romero Alvarez today on the Zoom, but is in fact Lindsay McCulloch. And her question for her comes from Mia T. And she asked, what are some of the experiments you are using to study the symbiotic relationships of soil microbes?
Sure. So there are lots of different ways that we experiment or manipulate the environment to see how that might change how plants and microbes are interacting together. So one component we think is really important is light, because the more light a plant has, the more they can photosynthesize, and then they have more carbon or food to give to their symbiotic microbial partners. So one experiment that we did was we planted seedlings out in the forest in these gaps, but then we also built these little like nurseries or shade houses where we manipulated how much light the plants got to experimentally see if plants were growing in darker places. Did they invest less in these helpful microbes? So those are some ways that we might experimentally test how soil microbes and plants are working together.
Awesome. Great. So the next question, I think we're going to make this for Danielle. And this is a student question. And the question is, one of you guys mentioned how you were originally a doctor who turned onto this path. Do you think that your background knowledge in medicine helped fuel your passion?
Yes, I believe that usually people ask me why, if you are completely turned into research, you lost your time following a medical career in the university. And actually, I believe I have an advantage against all of my research peers, because by working in the clinical world, I was able to watch how that portion of knowledge work, and then I can make connections between that portion of the world and then the pure field microbiological worlds. And that is an advantage because I am able to actually translate information from one world to another world. So actually, I believe that currently, whenever you are able to cross fields and cross career and actually mix multidisciplinary research, you will have an edge in order to find cool hypotheses and to put together cool science.
That sounds great. Thank you so much, Daniel. I had a question here on our other list for Arindam. And Arindam, you mentioned region-specific information in terms of training students. So I wondered, in the textbooks, kind of your standard textbooks for a microbiology course, do you feel that there's currently a lack of region-specific information, or like the wrong region, or what's the problem that is kind of being solved by having these region-specific examples?
Well, one of the focus of India's major thrust in education is the implementation of NEP, or National Education Policy, which focuses on experiential learning. So this kind of module actually helps take your students to the field, explore the nuances, so they can make comparisons between one region with the other, which is not there in the textbooks. Definitely. So that adds value and gives a sense of experiential learning to the students. So it gives the context.
Thank you, and so this question is actually for Jorge, and so we're wondering, the student asks, can you walk us through an actual example of field work? As in, what samples are you collecting? What preparation are you doing? Things like that.
Sure. So I'm so like interested in insects, and actually I'm doing like my PhD like in entomology. But in the past, as a part of this like nitrogen fixing like project, I went to the field and I collect like pieces of the nest of termites, like in a specific termites that have this symbiosis with this like nitrogen fixing bacteria. I collect like 50 termites, 25 soldiers, and 25 workers. And my aim with these things, and using like isotopically like enriched gas, we incubate those like termites during 48 hours, and then by by then, like we measure like the nitrogen fixation rate for those like 48 hours to kind of like expand because this termite is super abundant, like in the tropics or at least at La Selva. So we were able to kind of like uh, measure like the kind of like the nitrogen inputs of these like termites nests and termites like bodies or even termites frass to the environment. So like we like in resume like we start like with field work like collecting like a specific samples, then we move that thing like to the lab to do like something more like control, and then like the analyze to get like the kind of like the raw data that where they're like using like I don't know R for example, we start like to get like the results. Yes. That's clear. Yeah.
Yeah.
Okay, perfect.
Thanks, Jorge Daniel. All right. This is we have I have one for Carlos here. So in your module, right, you talk about changes in land cover and the association with the occurrence of the virus. So why would something like the presence or absence of a disease be associated with changes in land cover? And just for the students who aren't ecologists, changes in land cover means, say, taking a forest and converting it over to a farmed field or something like that. Vice versa, but it's never that way. Changes in land cover. How does that lead to changes in disease risk?
Oh, that may be a better one for Danielle, actually. But that's one of the things that I like about this case study because it really connects the disease, the infectious disease agent with the environment and human activities. And there's a really great image in the science article that Danielle published with the encroachment of urban buildings and interaction between humans and the potential vectors and hosts. Danielle, help me with the land use.
Definitely. So the situation is that whenever an ecosystem is altered, then the interaction between the species living in that particular place is going to change. And in terms of diseases, this relationship usually involves vectors that used to, for example, feed on mammals, then finding another mammal very suitable for their own hunger. So then mosquitoes start biting humans instead of biting non-human primates. And then a lot of diseases follow this pattern. And for Oropouche virus, we found that the loss of vegetation can be incriminated with the presence of Oropouche fever outbreaks across South America. So the idea is that as long as you alter the ecosystem, then the midges that transmit Oropouche virus is going to start biting humans, and then you have the Oropouche fever outbreaks. And this happens with multiple landscapes. And the essence is the change in interactions among species when the landscape is altered.
So we have so many more questions that we can answer, and I know the time's getting short, but I want to squeeze in one like big picture question. There's been actually a couple questions about the same kind of thing. So anyone who wants to answer, this is your time to jump in. All right, but quick answers. So It's about how outcomes of the research affects all of us in society. So one example might be, to what extent are microbial symbioses responding to climate change? Can these interactions buffer climate impacts? Does pathogenicity seem to increase as environmental stability decreases? Do we even have enough baseline data to assess these questions? How are we all impacted by this stuff? So anybody who wants to jump in with that kind of answer.
I would like to jump in answering that particular question. So specifically the portion that mentioned, do we have enough baseline data to even assess these questions? So the baseline data is kind of lacking. We still need to get more information. And at the end, this is going to be pathogen dependent. So in 2016, we published a super cool study called declining prevalence of disease vectors under climate change. And we were, in that particular modeling exercise, we discovered that vectors, as the temperature increases, they, instead of living in the tropics, they are going to start living in the Andean region, because this was done in Ecuador as well. And if they start living there in the Andes, then the area occupied by the vectors is going to diminish. And if you diminish the distribution of the vectors, potentially you diminish the prevalence of the disease. So it was a cool modeling study, and this should be applied to all diseases and all vectors. That is why this should be understood as a call for more research.
Any other thoughts?
Me, I want to jump in like to this question because it's super nice. Because right now, actually, like for my first chapter, I'm working with Osmia lignaria, that is a bee. Osmia lignaria is like a solitary bee that they collect like pollen from different like plants, like to create like a pollen provision. And I'm working in a latitudinal gradient through California because there is a paper showing that higher temperatures, like the more like pathogenic like bacteria, like Arsenophonus, for example, is more prevalent in those like pollen provisions. And the effect of this bacteria is that they call like the sun killer bacteria, because just kill the males and affecting like the sex ratio of these like bees. And this is an univoltine species, so you have one offspring per year, and so this will affect super like, so this will have like a kind of like a bad impact in like the like populations of like bees. But this is kind of like an example how you can kind of mix like temperature and climate change and these kind of things with like microbes, or even bees, soil, or things like that.
And I would just add a quick positive note is that a lot of these mutualisms with microbes have evolved millions of years ago. So they've existed in lots of different kinds of climates. And so it's possible that they're going to help plants respond to changes in climates as the climate changes, or help plants grow or respond to disturbance. So not everything maybe will be bad with microbes as the climate changes. Maybe they're also going to be helpful in helping ecosystems recover or respond or be dynamic.
So important to have hope. And I think Jeff has one last question for everybody.
Oh, sorry, we were just off on the same page a little bit. Did anybody else want to jump in and say, what challenges did you have with implementing the online module and making it? What do you think was just a real quick answer. What was the single hardest thing that you had to do to make your online module?
I can go. So regarding the implementation part, I think the students need to have a lot of background about microbial ecology and how the module fits into that particular unit. So yeah, it took a couple of classes. So the entire module that is presented in the Learn Gala, it's very user friendly, and students can use mobile device to access the module. And it is broken into two sections, unit 1 to 5 in day 1, and the rest, the interpretation of graph in the second section, data interpretation. So it has been implemented very effectively in that way.
I think one challenge for me is when you're putting together a lesson, you're always thinking, who is your audience? who are your students. And so when you're creating something that is going to be available online, there's lots of different students or different classes that it might be used in. So trying to strike that balance of providing enough background information that would fit a broad swath of like possible learning opportunities, I think was what was challenging for me.
Thank you all. And we're going to squeeze in one last question because we like to make this so student focused. So, does anyone have advice for students? Maybe they want to get involved in field work. Maybe they want to get involved with research. Maybe they want to know about this next step of their career, or maybe they had a setback and they feel like, hey, I need to be able to move forward, even though I failed calculus or whatever it was. So, just any advice that you all have for students?
I will jump in and I will say, and this will be super general, but be happy, and choose something that you feel really passionate about, because these like science is already so hard to like be working in something that you don't like. And I will say that choose something that you feel passionate about, feel I don't know. Feel free to ask like for guidance to the people that is where you want to be. I don't know, like any advisors or professors, but yeah, kind of like follow your heart in that sense. Follow and be happy. Yes.
I would like to follow that particular advice, mentioning that you should just literally jump in and do it, because there is so many information right now. With the revolution of AI, you have R in your hands. You are no longer like, oh my God, I don't know how to program, or oh my God, my mentor cannot help me. It's like, I mean, do it. Like, follow your curiosity. The curiosity should be your north star in order to find interesting questions and actually do stuff to answer them.
Yeah, and I think doing science is not a very straight path. So you hear something interesting, try it out. It's okay if you don't like it. It's okay if you're not good at it at first. I like failed my first plant ecology class and like I'm now a plant ecologist. So I think failure is a part of science, and so we learn and grow from it. And so that's totally normal. And I think if you lead with passion, like Jorge and Daniel suggested, then there's so many opportunities. So just bring your curiosity to the table and you'll have success and fun in science.
I can add a little bit a famous quote by Jane Goodall. She said, "You cannot get through a single day without having an impact on the world around you. What do you makes a difference, and you have to decide what kind of difference you want to make."
All right, now that wasn't fair to end with a Jane Goodall quote. That was low. I'm still feeling that one. All right. Um, I want to thank all of our presenters for being part of this today. Lindsay McCullough, Jorge Morasi Baja, Carlos Galler, Daniel Romero Alvarez, and Aranda Mitra. We also want to thank the Organization of Biological Field Stations, the Ocelots Network, the Center for Environmental Inquiry at Sonoma State University, and the National Science Foundation, who have all supported this enterprise in various ways. Don't forget to learn about all the amazing things that all these organizations do. And to help you do that, Carrie is currently putting a bunch of stuff in the chat right now that you can go and click those links and follow away to your heart's desire and learn everything you want to know about those guys. So like Daniel said, jump in, do it. We're going to be emailing all of you the next few days with follow-up answers to some of the questions that we didn't get to today, a link to a recording of the event, information and links to the student and faculty surveys that we talked about. Just a reminder, the faculty survey is a two-step process, starts with a consent form, and then we have to send you the actual survey. Thanks in advance for doing this. It's very important for us to track and assess the success of these events and making it better. So we hope to see you at future Live from the Field events. One on the topic of global change is coming up, 10 a.m. Pacific, 1 p.m. Eastern. That's Monday, November 17th. And we have another that's yet to have its schedule finalized in spring 2026 that will be about the chemistry of tropical forest biology. So we'll get this information to you, and please share the links with fellow instructors, students, friends, and colleagues. And so I guess now we say goodbye from the field. So panelists, could you wave goodbye to our student attendees?
And
Thanks.
yes, everybody, if you have more questions, feel free to email them to me. I put that email in the chat just now, and everybody here is going to answer those questions and we'll email them out to you. Thanks, everybody. Have a great rest of your day.
Thank you very
Bye.
much.
Thank you.
Bye.